Yeah.
Good.
Yeah.
So it's my pleasure, I guess, to introduce Emily today. So Emily is a new professor. How long have you been at Queen's now?
Oh, 9 months.
9 months into
Yeah.
her faculty position at Queen's. So she's taking, charge of the legacy of primate neurophysiology at Queen's, where there's lots of exciting opportunities. They have a very large scale program for macaques in particular. So yeah, so Emily did her PhD at Northwestern and then a postdoc at Pitt. And one thing about Emily's research that's really cool that she's developed over the years, of course, she's interested in brain-machine interfaces proper, meaning, tools to read out information from the brain and to reanimate things, but also is one of the people that is really using brain-machine interfaces as an experimental opportunity to probe particular aspects of the brain, like as a paradigm itself for scientific research. And I think having that combination of things is really exciting. And this paper, this work that I guess she'll be talking about today, I think is a just an absolutely beautiful example of that. So thanks, Emily, for doing this, and yeah, take it away.
Yeah, cool. Thank you for the invitation. Um, I have presented this work in sort of some more formal conference presentations recently. So I'm hoping to make this a little more casual and informal. So I invite you to interrupt me with questions, comments, concerns throughout, and make it a little more of a discussion. But yeah, so I'm going to talk about this recent paper. I think it came out in February, maybe. Dynamical constraints on neural population activity. Um, so let me make sure I can move my slides. Okay, there we go. So the idea here is based a little bit in this computation through dynamics framework. So in network models, the time evolution of activity or dynamics is shaped by the network's connectivity and also inputs into the network. And the dynamics that come out of that give rise to the computations being performed by the network and are often characterized by flow fields. And the specific flow fields here, like a point attractor or line attractor, reflect the specific computations being performed by the network. Oh, why are you not? Oops, sorry, too fast. So we've seen examples of this in the brain as well, where the time courses of neural activity in the brain also appear to follow a flow field in a way that resembles the activity that we see in neural network models. And these neural dynamics are believed to underlie computations involved in everything from motor function and sensory perception to decision making and memory. So this is sort of suggestive that computation through dynamics is a good framework for how things work in the brain. And a key prediction from this framework is that in the absence of external inputs, where you are in the landscape determines where you go next. So if we take a super contrived example here of the landscape is a hill and we have a ball, we sort of in the absence of external inputs, we might see the ball roll downhill, but without sort of kicking the ball, we're not going to see the ball like spontaneously roll uphill. So we wanted to test whether this fundamental aspect of computation through dynamics also applies to the biological networks of neurons. And we did this by asking to what extent neural population activity is constrained to follow specific time courses. Or in other words, are there also constraints on the time course of neural population activity? So the way that we answered this question, as Andrew alluded to, is by using a brain-computer interface. So I'm going to start by just describing the particulars of the brain-computer interface that we used here. And then I'll show you some examples of the temporal structure we saw or the dynamics that we saw in our neural population data. And then we'll try to answer the question of how flexible are these dynamics or this temporal structure. Okay, so we're going to start by considering just a simplified network of 3 neurons, and we can visualize the firing rates of those neurons as spikes in a raster, or we can visualize the firing rates of the population as a whole in the so-called neural state space, where each axis here is defined by the firing rate of one of the recorded neurons. And a point in this space represents the firing rate of our population during some short time bin. And this is what I mean when I say population activity pattern or neural activity pattern. And I use those interchangeably, so I apologize for that, but just to be super clear up front, that's what I mean. So over some period of time or during some task, we can plot the natural patterns of neural activity in this way. And many groups, including ours, have observed that neural population recordings exhibit highly structured, low-dimensional activity. And we can apply a dynamics view to that neural activity and connect these patterns in time to form some neural trajectory. So we might observe a trajectory like this. And if the time courses of neural activity are flexible, then we might see those same neural activity patterns generated in a different order to produce the time reverse trajectory. So this is what we want to test. If the activity time courses that we observe in the brain reflect the same sort of principles that we observe in the neural network models, then our hypothesis here is that the neural activity time courses should be robust and difficult to violate. So if this is true, then it should be easy for the monkey to produce this neural activity or neural trajectory because it follows the flow field set up by the underlying connectivity of the network. But it should be difficult to produce these same neural activity patterns in a different order because now that this trajectory violates the flow field. Yeah, okay, so that's sort of the hypothesis we're testing here. And to directly sort of test the flexibility of the neural activity time courses, we're going to use a brain-computer interface, or a BCI. In our BCI, we record with Blackrock arrays, so we have on the order of 100 neurons, and those neurons are from primary and or dorsal premotor cortex. We apply dimensionality reduction to that neural activity to find 10 latent dimensions. In this case, our dimensionality reduction approach is GPFA. And once we have our 10D latent space, then we find a 2D projection of that 10D space and provide the monkey feedback of his neural activity in this 2D projection as cursor position. So just to be super clear here, we go from about 100 neurons to a 10D latent space to 2D cursor position. And this BCI yields what we call an intuitive mapping, meaning that it's easier intuitive for the monkey to use. So a lot of our previous work is sort of BCI learning, and this is not learning, right? This is something that's very natural for the monkeys to use. They think about moving to the right and the cursor moves to the right. They think about moving to the left and the cursor moves to the left. And not sure this is necessary for this group, but some groups need me to say that the monkey is not moving his arm at all, right? He's only driving the cursor by modulating his neural activity.
Emily, question. So I'm curious why you chose position as the control variable. I mean, monkey thinking moving to the left could also have been in velocity terms or some other terms.
Yeah.
And whether do you think that impacts the setup of this question as a whole, meaning the flow field, for example, in those coordinates?
Yeah, so we think of the position mapping as giving the animal like a pretty direct readout of their neural activity.
Mm.
And so that allows us to request that they produce activity patterns in a particular order. so let me just walk through the schematic and flag me down if you want to talk about it more.
Yeah.
But yeah, so with this sort of feature, when we're mapping neural activity to cursor position, we can basically say like, okay, this blue neural activity pattern here maps to this blue position on the monitor. This purple neural activity pattern maps to the purple position on the monitor. Can you guys see my cursor?
Yeah.
Yeah.
Okay, cool. And this red neural activity pattern here maps to this red position on the monitor. So now, in our BCI task with this position mapping, if we want the monkey to produce this blue cursor trajectory, we can present, or sorry, if we want him to produce the blue neural trajectory, we can present him with this series of targets on the screen of blue, purple, red. Mm-hmm. And in order to move the BCI cursor along that trajectory, he has to produce the corresponding neural patterns of blue, purple, red. So using a position mapping as opposed to a velocity mapping gives us like very precise control over like where neural activity is and where we want to place it.
Mm-hmm, mm-hmm. Yeah, but I mean, I guess so how would the dimensions of neural activity, just look at the neural data in this position mapping scheme, compared to what happened if the monkey, let's just say, reached between those targets? Well, how do these relate? Are these closely related? Are these dramatically different parts of the neural space? I'm just wondering whether, I guess the sharpest version of that is to say, are you probing something at the edges of the experience of primary motor cortex, or is it kind of operating in its normal mode when it's doing this task?
Yeah, so we think it is tapping into like how motor cortex normally works. And a sort of next step in this work is to relate this sort of BCI mapping to actual hand movements. And in that work, we're going to show that this BCI it basically perturbs the neural activity along a task relevant dimension. So it can move the neural activity in some like dimension that separates reach plan between like right and left reaches, for example. So we do think it's the dimensions that we're working with here are highly relevant to reaching.
Okay, got it. Thank you.
Emily, can I ask a quick behavior question?
Yeah.
I was just wondering, once the monkey fully learned the BCI, what's the monkey is doing with the hand? Is it just resting there with 0 EMG at the rest position, or is it just actually contracting or moving or something like that?
Yeah, so he is nominally just resting there. Again, in this sort of work that's going to come out that follows onto this, we have the monkey actually reaching and doing BCI. So we train him to sort of like hold a touch bar, or it's a force bar. It measures forces. So during the BCI, he needs to just rest there. And if he exerts force on the bar at all, it will fail the trial. So he is trained to just kind of sit there. In conditions where we don't like train that behavior in, the monkeys typically do just once they understand what the BCI is, they will just sit there, and if the intuitive mapping is in fact intuitive and the cursor is doing what they expect, they're not moving their arm very much. In a learning scenario, if we perturb that mapping in a way where now the cursor isn't doing what the monkey expects, we do see the monkey tries to like move his arm in different ways to try to get the cursor to do what he wants. They usually figure out like, oh, that's not super effective, but I don't think it stops them from doing that.
And just to piggyback on that question, I don't know how big the BCI is or where you're recording from, but would you also be getting information from the saccades the monkey is making as the cursor appears or as the position appears where they have to move to? And could that information be a part of your neural data?
Probably not. No, we have, it's just it's either a 96 channel array in primary motor cortex, or a couple of the monkeys had two 64 channel arrays, one in arm area of primary motor cortex and one in dorsal premotor cortex. So we're not getting saccade information.
Makes sense. Thank you.
Yeah. Okay. So once we have this intuitive position mapping, now we can test how flexible these neural trajectories are when they're under volitional control. And the way we do that is to basically ask if the monkey can produce these same neural activity patterns in a different order. In this case, we're asking, can the monkey produce them in the red, purple, blue order? You can take this one step further. You can ask, like, can the monkeys learn to generate any neural trajectory from patterns within their neural repertoire? But for the purposes of this work, we're going to focus just on sort of the time reverse trajectories. So blue, purple, red versus red, purple, blue. All right, so this is our main tool that we're going to use. And when we look at actual data, you can see that with an intuitive position mapping that the monkey produces relatively straightable cursor trajectories. And I'm focusing just on two targets here. With the intuitive mapping, the monkey can move to 8 targets in a sort of center out sort of way. But if we focus just on this pair of targets, you'll notice that the trajectories, the blue trajectories and the red trajectories, are highly overlapping. And this overlapping nature suggests that the same population activity patterns can be recruited in any order. So just to be super clear about this, if we focus on this arbitrary purple position, you'll see movements from this position towards the blue target and movements from this same position towards the red target. So this suggests that the population activity patterns can be recruited in the red, purple, blue order and also the blue, purple, red order. This would indicate flexible dynamics. But remember that this is just one 2D projection of our 10D neural activity. And if we look at other projections of our 10D space, we see some interesting structure emerge. So this is our evidence of temporal structure, and we're going to dive a little deeper into this. So this is just another set of cursor trajectories with the intuitive mapping projection. And for those of you who read the paper, we call this slightly differently in the paper. We call it the movement intention mapping or projection. It's the same. We were just trying to be a little careful with our terminology there, but it's same projection. And just to orient you to the plotting format here, I'm going to indicate the intuitive mapping projection with a gray background, and this black outline is going to indicate that this is the projection that the monkey is receiving feedback in for the BCI control, or the projection that's viewed by the monkey during BCI control. So again, we can look at the neural activity that produced these cursor trajectories in other dimensions. And when we do that, we find projections for which the neural trajectories are distinct as the monkey moves between the red and the blue target. So rather than being highly overlapping, we see that the blue trajectories are separate from the red trajectories. And in particular, we find a special projection that shows strong direction-dependent trajectory curvature. We call this projection the separation maximizing projection, and we can go into the details if you like, but essentially we're trying to maximize the separation sort of at the middle of the trajectories here. And we maximize that while minimizing the variance around a particular cursor direction. Going forward, I'm going to indicate this separation maximizing projection with a light blue background. And the lack of a black outline here means that this is a projection that is unseen by the monkey. And the structure that we see here is suggestive that the neural trajectories are maybe less flexible than we thought when we were just looking at these highly overlapping trajectories in the intuitive mapping projection. So our question here is how flexible is this structure? How flexible are the neural dynamics? And can monkeys learn to change this temporal structure? So again, we're going to leverage our BCI paradigm to answer this question. So here I'm showing just a 3D representation of our neural trajectories and some corresponding 2D projections of them. So the structure of our neural trajectories exists in this higher dimensional space, but we can only show the monkey a 2D projection on the monitor, right? And the cool thing here is that we as the experimenters get to choose which 2D projection we show to the monkey. And so for the next experiment, we're actually going to let him look at the separation maximizing projection where we saw this cool temporal structure. So instead of having the monkey perform the BCI task using feedback in the intuitive projection as depicted here, we're essentially going to rotate our high dimensional space so that now the monkey sees his neural activity in the separation maximizing projection. So in this experiment, we're essentially giving him visual feedback of his neural activity in this projection where we observe this strong direction-dependent trajectory curvature. And now we can ask if that structure is flexible or obligatory. So if the temporal structure is obligatory, if the dynamics are constrained, then we would expect that the cursor trajectories that we see here are, if it's obligatory, we should expect that the cursor trajectories match the direction-dependent curvature that we saw in this projection when it was unseen by the monkey. So the temporal structure would persist. If the temporal structure is flexible, then we might see the animal straighten out the cursor trajectories. So this is sometimes takes some convincing of why this is a reasonable hypothesis, but we've seen a preference for straight cursor trajectories in human and monkey experiments before, where like subjects will do some like crazy things with their arms if it makes the cursor move straight on the monitor.
So the BCI at this moment is locked down. The BCI mapping is locked down when you switch from one projection to the other.
Yes.
Yeah. Okay.
Yes. So if the monkey, the mapping is the same. And if the monkey just kept doing what he was doing before, in this view, it would look curved to the monkey. Mm-hmm. If he doesn't like that it's curved and he has this inclination and he's able to modify his neural trajectories, he might do that in a way that straightens the cursor up.
Curious, I mean, I should remember that from the paper, but at this moment I don't. Did you reward the monkey on straightness, or is that just something that that you hope is implicit in their brain?
Yeah, in this version of the experiment, we did not. He just, as long as he got the cursor to the target, he got a reward.
Got a reward, okay.
In later experiments, we impose this boundary that tries to, it's a way of rewarding straightness.
Mm-hmm, mm-hmm. I remember now, yeah.
Yeah, so this is, not a very stringent test of flexibility. But yeah, so what we saw is that when the monkey is doing the BCI task and he's getting feedback in this separation maximizing projection, is the cursor trajectory showed this strong direction-dependent curvature. And the structure that we saw here looked remarkably like that curvature we saw in the same projection when it was unseen by them. So just to be super clear here, these sets of trajectories come from different trials, right? the trajectories on the left come from BCI trials in which the monkey is getting feedback in this separation maximizing projection. But this set of trajectories is coming from trials, a different set of trials in which the monkey was receiving feedback in the intuitive mapping projection. And we're just looking at the neural activity in the separation maximizing projection. So this is just one example, but we did this 100 times across 3 different monkeys, and in not a single session did we see the trajectory straighten out. So just, this is obviously not all 100 experiments, but just three from each of the monkeys. Yeah, but as we just said, like, this isn't a super stringent test. It kind of relies on, you know, these particular monkeys maybe not liking curved trajectories or caring about that at all, and then also being able to flexibly control the evolution, the neural trajectories. So in order to more directly test the temporal structure, we did another set of experiments in which we challenged the monkey to move backwards against the empirically derived flow field. So these little arrows here are the actual flow field that we calculate from the data. And the big arrows are just showing like the general flow. I'm going to include the big red and blue arrows going forward. I just want to be super clear that the monkey did not see those. That's just to help us interpret the data. So essentially what we asked the monkey to do is produce this time reverse neural trajectory. So we saw him produce something like blue, purple, red, and now we want to ask him to produce the red, purple, blue version. And the way we did that was to place a target along one arm of the flow field. So here, the blue flow field. And this intermediate black target is one that was easily reached from the blue starting target. We don't show any of that data, but he did do trials there, and he was basically 100% successful at reaching this target from the blue starting position. But to reach that black target from the red starting position would require the monkey to do something different than he was doing before, right? Like if he just kept doing what he was doing before, he would follow this red flow. And what we hope is that that different thing that he would do would be to generate the time reverse trajectory. But this is what we saw. So he didn't generate the time reverse trajectory, right? We don't see any of the black trajectories moving backwards against that blue arrow. Instead, what we see is that the cursor essentially continues to follow the natural flow outlined by the red arrow, and then would hook back to acquire the target when he got it. So we quantified this effect by calculating the initial angle of the trajectories and comparing this initial angle to the initial angle of the red trajectory here, which is the one the monkey produced when he was just moving from the red target to the blue target. And we calculate this initial angle relative to the direct path, which is essentially the time reverse trajectory, slightly different, but qualitatively it doesn't matter exactly what we reference to, but we decided to reference this direct path for simplicity. And so we're going to calculate the difference in initial angle between these two trajectories. So a value of 0 here means no change. The initial angles are exactly the same. This would indicate that the trajectories are highly constrained. And a value of 100 here means the black trajectory basically moves along the direct path or the time reverse trajectory, and that would be sort of maximal flexibility. And here's what we see. Over 50 experiments in our 3 monkeys, we found that the initial angle doesn't change very much. So this is not a significant difference from 0. So this task did not lead the monkey to generate the time reverse trajectory, but again, this task did not require him to do so for success, right? As long as he got the cursor to the target, he got his reward. And yeah, so maybe he just wasn't motivated enough to generate the time reverse trajectory. So as sort of a final test here, and to really see if the monkeys could learn to produce the time reverse trajectory, we constrained the acceptable path to the target by imposing this visual boundary. So now, to successfully complete the task, the animal had to move the cursor to the target without exiting the boundary. And again, just to be super clear here, the monkeys do not see the red and the blue arrows on the screen, but they do see the targets and they do see this black boundary. And we start these experiments with a quite large boundary, one that we think should minimally affect the cursor trajectories that he showed in the unconstrained condition. And when we do that, we see high success rates and we see similarly curved trajectories. So the mere presence of a boundary didn't like suddenly inspire the monkey like, oh, wait, I can't just do whatever I want. Maybe I should go directly to the target. That surprisingly didn't happen. Okay, now for simplicity here, I'm going to show just the average trajectories for successful trials as we gradually reduce the size of the boundary. And what you can see is that over the short term of about 500 trials, the trajectories don't change very much. And I just want to point out here that the success rate is dropping as we decrease the size of the boundary. So rather than changing the initial angle of his cursor in a way that would allow him to continue to complete the task with high success, the monkey's only minimally modifying his trajectories. And that means for the smallest boundary here, he's failing about half the time. So given this behavioral pressure, he should have been highly motivated to straighten out his trajectories if that was within his wheelhouse. Just to give you a slightly higher level view of what an experimental session looks like, along the top here, I'm showing some individual trials in the light gray, and the average. This is average of successful and unsuccessful trials in the black as we decrease the size of the boundary. And you can see how that is affecting the success rate along the bottom here. So dashed line indicates when we changed the size of the boundary, and this required him to have some like minimal level of success, 75% success or higher, before we would make it harder for him. So we got to some point here where he was really struggling. So it's a really fun experiment to run, not frustrating though. Yeah. Okay. So just to highlight here, the small changes that we see in the cursor trajectories around here still basically follow the flow field that we initially observed. And again,
Can I interrupt?
Yeah.
I have a question. So I was thinking if you see any relation between the velocity of the cursor and the curvatures that you see, for example, in this constraint version, are the trials that are successful? Because I was thinking maybe if for example, in the unconstrained version, maybe on the trials that there's a bigger curve, the curvature has a relation to the speed of the cursor. So for example, in this constraint version, maybe the correct trials are performed more slowly. Do you see something like that? Or?
Yeah, it's a great question. We don't see a difference there. Let me pull up a slightly different figure here. Jumping ahead a little bit. This is just multiple examples of trials where we see what I want to point out here is the different levels of curvature in the trajectory. So we see some that are like quite curved and some that are more straight. And we did have a hypothesis early on that like, oh, maybe the ones that are more curved, he's more likely to be able to adjust those or something like that. We didn't find that to be the case. So that's one thing I wanted to mention. Another thing is that the position mappings are they're quite fast. So because they're a pretty direct readout of the neural activity, the monkey can get like pretty close to the target in like 1 or 2 time steps, which is different from maybe a velocity decoder where there's some like dynamics sort of built in. We typically use a velocity Kalman filter in our other BCI work. So there's some like dynamics there, and you know things are gonna kind of move smoothly, and there's like a little more control over the velocity. But the position mapping is quite fast. And I think that's why you see this sort of like, he gets pretty close there and then he has to like hook in to get the target.
Quick question. So this suggests that the neural activity, the temporal neural activity is constrained after the training. But if you took a different stimuli and tried to influence this during the training before the neural dynamics are set, do you think you would have some level of plasticity where you can change this?
Yeah, I guess I'm not sure what you mean by the dynamics being set.
So before the curve is created, before the monkey learns the task, if you had a different monkey who hadn't yet learned to do the task, and if you tried to impose the boundaries on them right now, do you think they'd be more successful in learning a different way to do this?
Yeah, I don't think so. Just to point out that like in the intuitive mapping, the monkeys can move flexibly in that space. And this is just a different view of that same neural activity. And he can't flexibly control this space.
I think the explanation, the intuitive mapping comes from a particular projection of neural activity, which is by definition doing what Emily's, I mean, she found the neural mapping that does that. And then found another neural mapping, which she defines differently, which is the one that has the maximal separation between them. So I don't think that that's not really a handle she has on the data in this approach. But I had a question for you, Emily, that was related. How special is the separation subspace that you're identifying, right? I mean, it kind of experimentally very good one to look at because by definition, it's the most separated. And it seems to be one where the inputs can't overcome the intrinsic dynamics. But one could imagine that, you could have a similar situation. I mean, that doesn't seem to happen by chance. That would be a bit unusual. But on the other hand, you could imagine that you have one that's similarly separate, but where the inputs can drive through the intrinsic dynamics. Do you ever explore that along those lines?
Yeah, so it's something that we're looking into. It doesn't seem like so what we have done is sort of looked in the other dimensions during this learning scenario. So is it the case that we see some like changes in the other dimensions where that would suggest like, oh, the monkey is trying to do something different and it's just like not making its way into the BCI.
Yeah.
And we don't see evidence of that. Occasionally we do, but primarily those changes are like in the intuitive mapping projection. So it's more that we think the intuitive mapping is a special projection. And
I see.
This one is just like, oh, it is special because we see maximal separation, but it's not special in terms of like, it's less constrained or more constrained than the other
And
projection.
just a quick follow-up to that. And so is what's happening in practice in this experiment, is this just shown for the animal, this particular mapping is then shown for the animal for an extended period, or do you occasionally have to show him that intuitive mapping to keep him calibrated somehow?
Yeah, so this is one experiment. So yeah, we only make it harder. We try to keep him at about 75% success.
Mhm.
I mean, this particular monkey was like willing to work at 50% success rates, but one of the other monkeys was spoiled, and if success rates dropped too low, he would just stop working.
Yeah.
So yeah,
Emily, this is Paul Gribble. Hi, nice to meet you.
Hi.
I have a question. Was it a different intuitive mapping each sort of session, or was it did each monkey have one?
Yeah, so we calibrate a different intuitive mapping each session.
Yeah.
We
think in a way, what it sounds like is within each session, the monkey learns something first to complete the task. And then within that session, from then on, they're kind of unwilling to change how they do things. But then presumably in the next session, is the intuitive mapping different than the previous session?
Yeah, the intuitive mapping is like pretty similar from day to day. And we've shown in other work that like if I calibrate an intuitive mapping on Monday,
yeah,
the monkey can like use that mapping for 2 weeks with no decrement in performance.
Yeah.
So it's the intuitive mapping doesn't change very much, but we do still yeah, we want to start with the best performance we can for the monkey. So we do calibrate it each day.
Right.
Just
It's quite a demand to put on a monkey after having it perform so successfully for a while to then try to do something different.
Yes. Yeah, so we try to not spend too many trials on the intuitive mapping.
Right.
We want the monkey to still be motivated by the time we get to the more challenging part of the day.
Yeah. Okay, thanks.
Can I ask a question, Ellie? Hi, I'm Ali. Nice to meet you. Based on what you said, for every 2D projection of that 10-dimensional, there would be some sort of weights that would look straight on that 2D projection based on the activity of neurons, is what I'm understanding. So every 2D projection could be intuitive if you change the weights in a way that the movements look straight. And then when you start looking with the same exact weight, locking the weight, when you start looking at another projection from 10D to another 2D, with those weights, it wouldn't look straight. And when you find a maximally curved one, it looks like now you're looking at the least intuitive mapping possible between those 10 neurons to some sort of 2D projection. So is it similar? So is this task showing something similar to, for example, you ask someone to like move your hand forward, and then suddenly you ask them, okay, now play tennis. Is that some sort of the neural substrate behind that kind of behavior? Or because you call it intuitive mapping after changing the projection, but now it's a totally separate projection. So it seems like now you change, now you have to, if you want to make it intuitive now you have to change the weights of your BCI, and now you can make it in a way that it looks straight. So for example, would there be an intuitive mapping between a 10-dimensional neural space and a 10-dimensional effector space, like 10 bars moving up and down?
Yeah, I mean, maybe like we call it intuitive because it reflects the intuitive movement intentions of the monkey, right? Like before we even train a BCI, the monkey is trained on a reaching task. So he knows, oh, when I move my arm to the right, the cursor moves to the right and I get a reward, right? So he's like overtrained on that. So he has some movement intentions in his mind, like we all do. And that's what I think the intuitive BCI mapping is capturing, right? Like he wants to move to the right. We come up with the algorithm that does that. And that algorithm is basically staying the same here. And we're just giving him a different view of his neural activity in the separation maximizing projection.
Yeah, so my question is, in the other view, the separation maximizing projection, could you change the weights of your BCI system that takes the neural data into movement in a way that now it looks straight instead of curved? Now it looks intuitive again. So what I'm saying is that it's like you're probing the neurons to do some new novel skill, but you're not like, now you're making them to like completely, it's like just basically like learning a new motor skill, for example.
Yeah.
To me,
Yeah.
it's like,
Yeah.
the mapping being intuitive again, because now it's non-intuitive for the same group of 10 neurons.
Yeah. I think that should be possible, but I don't think it's a straightforward, in terms of the feedback that we would give to the monkey, it's not straightforward exactly what that should be. Or exactly, we have to like reverse engineer what that mapping should be.
Emily,
But it should be possible, yeah.
follow-up question on that. Well, you know, one of the most, I think the interesting conclusion from your work here is that the monkeys seem to be really inflexible in changing their, you know, 10D neural activity. But of course, as you said, we know that in response to strange visual motor transformations like joint space to hand space or force fields or visual motor rotations, monkeys can learn to do very different things at the motoric output level to generate the same visual hand path. So how come, can you reconcile those two things? How come that's not happening here?
Yeah, I mean, it's a good question. I think it's just a constraint on how the neural activity can change. And I'll just emphasize that this is like a short-term learning experience, right? In our other work, we've shown, okay, within the manifold, the monkeys can learn to generate patterns within the manifold in different ways within a single session. More
it kind of goes to Ali's point that the test you're giving them here is kind of the most extreme version. Can you generate something way, you know, maximally different than the pattern that you've adopted? So I don't know the real world motor skill that will be. It will be something even more complicated than tennis. It would be, I don't know what, but
yeah. So what's interesting here is that the neural activity that's required to do this task, we know the monkeys can generate those specific neural activity patterns because we observed them, and all we want the monkey to do is generate them in a different order. So just going back to this thing, right? Like the monkey generated all these neural activity patterns. We know he can do it. We've seen him do it. He's done it multiple times. And all we want him to do is generate those same activity patterns, but in a different order. So this is a specific type of flexibility that we're testing here. It's not, can he do something new or generate new neural activity patterns? We're not asking him to do that. We're asking him to,
well, it'd be like me asking you to, if I asked you to sign your name backwards. Yeah. And I said, oh, well, it's the same hand positions. You've done this before. I'm just asking you to do them backwards. Yeah. It's not necessarily, it's a little bit of a straw man, I would say. It's not so obvious that it ought to be possible.
Yeah. But I think this is part of part of the point, right? Is
Yeah,
like
I think that's right.
the computation through dynamics framework says like, oh, it matters how the neurons are connected.
True, yeah.
And so this is in support of that, I think. And I mean, there's two things, right? We are really testing the flexibility of the recurrent dynamics, like what is intrinsic to the connectivity of the network. In this set of experiments, the monkeys had every opportunity to change their inputs however they wanted to. And we tried to guide them a little bit with our tubes. There are boundaries. But at least in the short term, the inputs weren't able to sort of overcome the dynamics of the connectivity of the network.
Yeah.
So
It's
yeah,
different than, I mean, I think it's just to Ali's point, this is different than learning a whole new behavior per se. This is the mapping within the neural population. Clearly, you can change other parts of the neural population to achieve some new task you want to learn.
Yeah.
But you can't arbitrarily associate patterns of neural activity. It didn't have to be that way, but that's what this suggests, right?
Yeah.
Can I ask a question? I think it is related. So I was thinking, now that you're looking at the separation maximizing, you haven't done this year, a dimension. What do you think about, for example, the dimensions that are agnostic to the movement? For example, the two targets on top of each other. Do you think you're going to see the same unflexibility there? For example, you can't induce dynamicism inside those planes, for example. For example, you have planes that the two targets are always on top of each other, right? In this task. Do you think that monkey would be available to dissociate them? I mean, I'm trying to see how unflexible the system is. So if you look at it from different dimensions, would it always be that unflexible as it is in this dimension, you think, or not?
yeah. So we have primarily just looked at these dimensions. But when we look at the other dimensions that aren't the BCI feedback dimensions, we see similar levels of flexibility, meaning they're not that flexible. The exception to that is the intuitive mapping projection, which is special in some way. And I mean, it's special because of how we calibrate it and what it needs to do. It almost has to have those features, but
yeah, because the main reason I asked was that maybe the other dimensions that nothing is happening basically in the, for example, in a dimension that the 2 points are on top of each other, I was thinking maybe it has more flexibility recruiting those dimensions if needed. I don't know what would happen there. You know what I mean?
yeah, I don't think we see evidence of that. We did, if anyone delved into the massive supplementary figures, this was our attempt to sort of look at that. And what we did was in D through H here is we took we characterized these flow fields in a bajillion different 2D projections and then tested well, how different are they? Are they changing in the other dimensions and we're just not capturing it? And we don't see that. We see that things are pretty constrained. And what we compare, we have some comparisons here where things are quite different or quite similar.
Yeah, and I meant in another task. I know in your task you don't see it. I thought if you target those dimensions specifically to be recruited with UC, for example.
Oh,
you don't have, you haven't done the experiment, but I thought you may have some intuitions there.
I'm sorry, I see. So you're saying in other tasks are the separation maximizing projection dimensions more flexible?
No, for example, if you have a task that instead of targeting the separation maximizing projection, you have that projection that the targets on top of each other. There's no dynamics inside the task. Okay, for example, you had this third dimension that you showed in your figure that nothing is changing, right? Other than the intuitive mapping and other than the intuitive dimension and the separation maximizing dimension.
Yeah.
You enforce like sort of a separation of the targets in another task, you think, to see if they can change that dimension, for example, that nothing is changing there.
Yeah, I see. So yeah, I mean, I think probably not. And I say that because here, when we're like testing the flexibility, we focus on just one pair of targets, but we have 4 pairs of targets, and we can we only test one pair of targets in a given experiment, but we tested them all for all the monkeys, and we essentially see that there is some like underlying structure of the dynamics that is pretty constrained from task to task or from day to day.
Thanks.
Thanks.
I have another question. If you have time.
Yeah, we have.
Okay.
So I was curious about the nature of this intuitive mapping. So you said you put the electrodes in the arm areas of the motor cortex. So for example, if you put the electrode in like on a hand area or like finger areas or foot areas, eyes, other areas, face areas, could you in the same position task, could you find an intuitive mapping between the neurons in those areas, or is it something specific about arm that like can reach?
Oh yeah, I mean, yeah, the intuitive mapping you can find, it might be slightly different, right? Like if you think about the hand, like there's a lot of degrees of freedom there, more than like the 3 of the arm, but or 3 of just moving the arm in space. So maybe it's slightly more complex, that intuitive mapping, but yeah, you can
Right.
generate intuitive mappings in a lot of different ways. The specifics of how they're intuitive, like, oh, maybe for the hand, there's like moving your thumb moves the cursor to the right and moving your pinky moves the cursor up or something like that, right?
So now, like, let's say you put it in monkey's hand area or finger area, and then you adjust the weights in a way that on a 2D space it looks straight. And if you could ask monkey and the monkey could talk, it would tell you that, oh, I'm like pressing thumb and index together and then pinky and thumb together. And now you change this projection, but you still keep the weights the same. And now the monkey has to do a really, if you could ask it again, it's like, I can't do it because I have to really weirdly move my finger and like, I don't know, do something completely new and unnatural. So talking to my previous point, do you think that would be the case that, for example, in the arm, now you're requiring monkey to like do something really weird with their arm in a sense of a neural space?
Yeah, I mean, potentially.
Which is difficult because of the structure of the neural network.
Yeah. Yeah, that might be the case, right? Like, but I think that has to do more with the structure of the neural network and what those neurons drive in terms of how the arm moves.
That's a tough one to dissociate, right? Because now you're kind of in an evolutionary, like,
Yeah.
are these constraints there because of the biomechanical constraints that are normally there or not? I think it's a good hypothesis you have that these patterns of coordination don't happen in nature, therefore you can't arbitrarily produce them. Yeah, but evidence for that is going to be limited, I would say.
Yeah. So my point is that even when you rotate, it is like requiring them to do completely new thing.
But that's the test. We want to see if they can do it.
Mapping thing. No, but for example, we know that then you can move your fingers. You cannot suddenly play guitar. You have to like train for a long time. So this task seems to be a parallel to this, but in a more controlled and
Well, but if the
No.
system were driven by an input, you could from some other area, then you could arbitrarily drive those inputs and make this go through any trajectory. And this is demonstrating that, well, at least insofar, at least in these local circumstances, there's a lot of constraints set by the intrinsic dynamics of the system, and you can't just overpower them.
Yeah.
Or at least the monkey is not sufficiently inclined to do so under this set of conditions. Is that a reasonable interpretation, Emily? Like that giving there?
Yeah, I think that's consistent with how we're thinking about things.
Mhm.
And yeah, I mean, these are single day experiments, right? And we have other BCI learning work that in order to learn something that we think is like the BCI correlate of a skill, it takes the monkeys a week or 2 weeks to learn to do that. It might be if we gave the monkey this task for 2 weeks, this, separation maximizing view, and we gave him a week to learn it, maybe he would learn how to do it. My personal hypothesis is that it will take even longer than learning an outside manifold perturbation.
Mm-hmm.
You're familiar with that work, but we have not done those experiments yet.
Mhm.
I think one point of confusion, just like in general, is this notion of intuitive. When you say intuitive mapping, maybe that's a bad word. Because I think it's like, it kind of, bad in the following sense, right? That it kind of suggests that it's intuitive to the monkey. Right? But it's not really that. You're like literally choosing these ones that go straight on the screen.
Yes.
I mean, that's what you're doing, right? If I understand it correctly. So it's intuitive insofar as that is the trajectory that the monkey generates when they would reach between these targets or look between those targets, right? But it's not intuitive in the sense that you turn on the BCI and say, okay, monkey, move the cursor right, and it kind of just produces them. If I understand that correctly, is that right?
Yeah, I mean, I think this varies a little bit with how one trains a monkey to do BCI. Because they're all overtrained on reaching tasks before they see a BCI task by necessity. But we also introduce the BCI in sort of an incremental way. And that likely influences this as well. But once the monkey gets it, he knows he's doing BCI, then I think the intuitive mapping is intuitive to him.
Yes.
Yeah.
It's intuitive to him, but yeah, okay. I think that is like the point, is like to whom is it intuitive when, I think is an interesting semantic thing, but I think it influences how, at least for me, how I can map through the set of experiments, basically, right? Yeah. Well, it's an hour in, and you can see that there's a ton of excitement about your work. I don't know, is this a good place to end, or do you want to go through the rest of it? Or I don't want to keep you longer than we've requested your time for, first and foremost. So.
Oh, we have gotten basically to the end.
Okay.
The last thing was just we quantified this change in initial angle in response to the boundary task. So we also find that it's highly constrained there. Very little change. Yeah.
Perfect. Okay. Well, thank you very much.
Yeah, cool. Thank you.
Yeah, we really appreciate it. Like you see, we're very excited about that paper. It's definitely stirred various conversations over the time. So we appreciate you taking us through it, and it's good to see you.
Yeah, cool. My pleasure. Thanks, guys.
Thank you.
Thank you, Emily.
Bye.
That's interesting.